Good morning, everyone, and welcome to our webinar on soil sampling principles and analysis methods. My name is Erica Yaskowicz, and I'm the Crops Extension Specialist with the Saskatchewan Ministry of Agriculture in Kindersley, Saskatchewan, and I'll be chairing today's event. CEUs are being offered for certified crop advisors on this webinar. To get credit, you need to attend the entire live event, and there will be a QR code at the end of the presentation to collect credit. If you missed the code and have attended live, please email us afterwards and we can work with you to get your credit. Before we start the formal presentation, I have a few items to share. Our team of crops extension specialists put on the homegrown webinar series each year. We work out of 10 different regional offices across the province that are represented on this map. Each office contains a wide variety of specialists who can help you with your agriculture-based questions. If you don't know the phone number of your nearest regional office, then you can contact the Agriculture Knowledge Center for all agriculture-related inquiries in Saskatchewan. The resource agents managing the Agriculture Knowledge Center call line will connect you with a specialist in your area to answer your inquiry. In addition to answering agriculture-related inquiries, the Agriculture Knowledge Center supports clients through the Saskatchewan Crop Reporting System, Provincial Rabies Hotline, the Sustainable Canadian Agriculture Partnership, Premises Identification, and Agricultural Crown Land Inquiries. Have you heard of Saskatchewan's AgriArm sites? AgriArm is a network of producer-directed applied research and demonstration organizations located across Saskatchewan. There are 8 AgriArm sites throughout the province that provide producers with data on sustainable farming practices and technology that is specific to Saskatchewan's different growing environments and soil types. Many of these sites host field days, and they take turns hosting crop diagnostic school each year. This year, Crop Diagnostic School will take place at the Northeast Agriculture Research Foundation by Melfort, Saskatchewan on July 24th and 25th. Keep watching our social media pages for registration details in the coming months. Lastly, we want to ensure everyone is aware of the funding available through the Sustainable Canadian Agriculture Partnership. You can reach out to your local agriculture program specialist to find out what funding you may qualify for. For today's webinar, we have 2 great speakers presenting soil sampling principles and analysis methods. First up is Dr. Jeff Schoenau. Dr. Schoenau is a professor of soil fertility and a professional agrologist who works in the Department of Soil Science at the University of Saskatchewan. He holds the Saskatchewan Ministry of Agriculture Soil Nutrient Management Chair in the College of Agriculture and Bioresources. He was born in Saskatchewan, completed his undergraduate and graduate degrees in the 1980s in the College of Agriculture at the University of Saskatchewan and has worked there since. His research, teaching, and extension activities deal with soil fertility and fertilizers, nutrient cycling, and soil management practices in prairie cropping systems. The second speaker we have today is Dr. Derek Peak. Dr. Peak is a professor of environmental soil chemistry at the University of Saskatchewan with 25 years experience performing spectroscopic analysis of soils using infrared and synchrotron techniques. Dr. Peak's soil chemistry research group focuses upon linking chemical speciation of nutrients and contaminants with their fate, biological activity, and transport properties in the environment. As a result of the importance of chemical speciation in natural systems, he has worked with a wide range of industrial and academic partners to use spectroscopic techniques to better manage contaminated sites and improve the efficiency in agronomic practices and the development of new analytical approaches for environmental and agricultural systems. In recognition of Dr. Peak's innovative work using spectroscopy in agricultural research, he was awarded both the College of Agriculture Bioresources Dean's Award for Research Excellence, as well as a University of Saskatchewan Global Research Leadership Award in 2019. We have a few housekeeping notes to keep in mind during the webinar. Each speaker will have a 45-minute presentation followed by a 15-minute question period after each speaker. If anybody has any questions during any of the presentations, you may type them in the question box in the bottom right-hand side of your control panel. Your question will remain anonymous and will only be sent to me. During the presentation, you'll be muted so that we can ensure there is no feedback to interfere with the audio. The entire session will be recorded, so if you miss anything or wish to revisit any of the presentations, you will be able to. Recording links will be sent out within the next few days in a follow-up email. An evaluation will pop up at the end of the webinar this morning. Please fill out the evaluation, as this helps us determine what topics you want to learn more about and help us prioritize research needs for the future. With that, we'll get started. So go ahead whenever you're ready, Jeff.
Okay. There we are. And I'm just trying to get on the webcam. There. All right. Perfect. All right. Trust that everybody can see my screen, and it's great to be here this morning. Thank you very much, Erica, to talk a little bit about soil sampling and analysis interpretation. So I'm going to mainly talk about wet chemistry and sampling, and Derek's going to follow up and talk about spectroscopy this morning. So I guess as a little bit of lead in, I think when we're thinking about the 4 R's and rates, we certainly realize that soil sampling and analysis is the foundation for decisions, good decisions made on rates of nutrient application. And we think about fertilizer recommendations for rate of applied nutrients. I think we think about those fertilizer recommendations. We consider them to be sound agronomic advice that's going to advance the benefits, the agronomic, economic, and environmental benefits from nutrient application. Certainly not gospel to be blindly followed. And really, it takes folks, knowledgeable folks, a lot of the folks in the audience today, I'm sure, are involved in crop consulting, really to help growers develop crop nutrition plans. And really that involves a number of steps, starting out with soil sampling of the field, followed by the soil analysis that becomes one of the bases used for fertilizer recommendations. And as we'll see, those recommendation tools that are in use out there do indeed vary in their sophistication, all the way from a sheet of paper with maybe 3 or 4 series of recommendations according to different target yields or yield potentials, all the way up to some fairly sophisticated hands-on decision support software. As a bit of background, I think before we get into that is to think about different approaches to nutrient management. We can break those down really into 3 categories. The first is sufficiency, and it's kind of looking at things in the short term. What do I need to add in the upcoming growing system, in the upcoming growing season, I should say, to ensure that we've got enough nutrient there, the right rate, so that yields are not limited in that present crop. But other approaches that we also may be thinking about when we're thinking about soil fertility management over the longer term would be a maintenance, where we would be looking at how much needs to be applied for that year, and also what are we going to apply to replace the nutrients that are harvested in the crop, to keep things in balance over time, to keep those plant nutrient levels at non-limiting levels and maintain our fertility. And some folks may even be looking at a build-up approach, where we add enough of the needed nutrient, and also looking at ways that we may build up our nutrient soil supply or fertility to a non-limiting level. And those kinds of maintenance and build-up strategies are most appropriate for nutrients like phosphorus and potassium, in which they're not easily lost from the system if we have more there that's have more there than what the crop requires and takes up that particular season. So how are fertilizer recommendations arrived at? It's kind of a sequence that goes on here, and really it starts out first out there in the field, getting that soil sample out of the field and analyzing it to measure the amount or the supply rate of available nutrient, what we might call collectively as a soil test. The second step, though, involves taking that information along with other information and essentially incorporating that into some type of a model where we have a prediction of environmental conditions, associated crop yield potential, and the crop nutrient demand. So we have to have that physical model then to use that information that's provided from our soil test. And we also need to be thinking as well about, of course, economics. And so within those models, or a separate model, we may be considering how much fertilizer do we need to apply such that we maximize the return, the net return on that fertilizer investment, maybe in applying economic principles like marginal cost versus marginal revenue, applying fertilizer up until the last dollar spent gives you a $1.50 maybe worth of increased yield. And then the fourth part of it all is kind of a human element, I would say, and that is understanding the grower and what their goals are, what their attitude to risk is. And really, it all starts with soil sampling and getting that sample out of the field at the appropriate time, handling it properly, and certainly, as we'll talk about, accounting for the variability that exists out there. And of course, a good crop nutrition fertilization plan is only going to be as good as the sample it's based on. And so we need to get the best representation as we can of the nutrient status as it exists out there in the field, and stress, really what we want to do is we want to understand and account for that variability. And variability, we're talking about spatial variability, variability across fields, among fields, even among regions, and then temporal variability. That's variability over time. And if we look at scales of spatial variability, we can look at it at different scales, all the way from regions, even across an entire province, literally several hundreds or even 1,000 kilometers. A good example, the soil climatic zones that we have here in Saskatchewan that represent differences in expected precipitation versus evapotranspiration, that also manifest itself in differences in things like organic matter that tie into things like mineralization and so on. Moving on to a smaller scale then, on an individual farm basis, we can have differences between fields, even within fields, that relate to past management, topography, parent material variations. And another source of variability that we can't forget about is what we term microscale variability. And this is variability that can exist in nutrient availability and soil properties over only a few centimeters in the field, that may be caused, for example, by things like residual fertilizer bans, and that necessitates a particular sampling strategy as well to account for that. For example, sampling across the seed rows, making sure that you've got enough cores taken so that you include both those areas within and between the seed rows in a representative fashion. And I guess, when you think about it, it maybe seems kind of random at first glance, but really, again, what we're trying to do is we're trying to convert that random variation into systematic variability, and that is variability that we understand and then can deal with. And that really involves, first off, understanding and recognizing, first off, that the properties of the soil, the yields, are different in different places for a reason or reasons. And then what we want to do is we want to group those according to the source and nature of the variation. And then that enables us to really create separate management units or zones within the field that becomes then the basis for a site-specific soil management, for example, a variable rate fertilization. So when it comes to developing or delineating those management zones, and there's a lot of different approaches, and the Ministry of Agriculture has a variable rate treatments fact sheet online that's a good source of information and background on this. We can do this in a number of different ways. Yield maps off the combines, some new technology now. Some of the combines out there also have protein maps, so both yield and protein maybe together, identifying an area of low protein that may be indicator of a low supply of nitrogen in that field, can be used to delineate areas that you would want to have as separate management zones that you would sample separately. We got lots of imagery out there. Aerial imagery, satellite imagery from drones, can be as simple as a black and white aerial photograph from the old days to NDVI imagery. Another, I think, very effective way to delineate management zones is according to topography, looking at the landscape and the surface curvature and how that influences water, nutrient availability, productivity across the landscape. We have our mapping that's now digitized, our SKSIS system that allows us to look at the soil association and properties from digitized soil survey information. Other approaches, things like looking at electrical conductivity across a field. And then, of course, we have our soil and plant sampling, that testing that may be done across a field on a grid type approach, for example, that would allow the creation of a map. Again, delineating those zones that we sample and manage separately. I know there's a good number of folks in the audience who have taken my Soil Science 312 class and gone on the sampling trip out to Goodale Farm, and we do a very simple topography or landscape-based sampling out there. This is a digital elevation map of that same site, and folks go out and sample in a directed sampling approach, the upslopes, the mid-slopes, the low slopes separately, and analyze those separately, and tells a lot about how the fertility varies across the field and the importance of landscape in controlling that. And I guess moving ahead a little bit beyond that, we can get even more sophisticated in terms of our delineation and division of the field into different zones or areas. This is the SKSIS predictive soil mapping, and this is going to be coming down the road where you can do use digital elevation maps to do slope position mapping and create some very detailed descriptions and portrayals of landform element complexes across farm field areas. A common question that comes up when we're talking about soil sampling is sampling depth. And oftentimes get the question, what depth is best? And it's not really a simple answer to this. It depends a lot on the nutrient, the cropping, the management history, and those depths are often a compromise. And so if you think of just taking a 0 to 24 inch depth, that's not really going to tell you something, but I think much more effective is actually the segmentation into different depths and taking a look at the distribution of nutrients with depth can be very rewarding. And overall, I think we can say that a shallower sampling, like an A horizon or a 0 to 6 inch type sample, or 0 to 4, that surface horizon is certainly, I think, probably better for immobile nutrients like phosphorus and potassium that don't move too far in the soil from where they are to the roots, and the bulk of the roots there are right there in the surface. We don't want to be diluting that sample too much with what we have at depth, where the roots aren't going to be big interactors with those nutrients, at least in the early stages when they need them. Micronutrients as well for some of those that are rather immobile. Organic matter content, mineralization supply of N, it's up in that surface horizon where a lot of that biological action takes place. Now, other nutrients like nitrate and sulfate, those are mobile. They can move, they'll move with the water from wherever the water's coming from to those roots, and nitrate, sulfate at depth can move with water to the roots later on in the season as the water is used from deeper in the profile. They're present at depth, they contribute to crop nutrition. It's a good idea to then check and account for those subsoil reserves as well. A good example in a lot of the soils in the drier region of the province, the dark brown soils do have significant accumulations of sulfate as gypsum and CKSA horizons within the root zone, and it's a good idea to check and determine the presence of those types of reserves. Generally, that deep nitrate, for example, becomes more of an issue if you've been fertilizing for a number of years, maybe have ended up putting on more than what the crops removed, combined with some wet conditions, moving that nitrate down deeper. You may have a reserve down there, and something that I think is important to know what exists at depth. I'll move on to soil analysis, and of course, after you get that sample out of the field, and sample handling is also very important. We want to preserve that soil in its state as much as possible until it gets into the lab, and so different approaches to that would be air drying or freezing. You don't want to leave it wet in the trunk of your car for 2 weeks. Things are going to change, especially for nitrogen. But once you get it in, the next step is some type of an analytical technique or soil test to extract or assess the nutrient availability in the soil sample. And that involves, depending on the process, extracting solutions. Another is PRS, plant root simulator, ion exchange resin membrane probe, as ways to assess the available nutrient in that sample. And those extractions that are used, for example, a calcium chloride extraction or weak salt solution for nitrate, what we would term water soluble, is essentially a kind of quantitative assessment of how much is there. But many of the extractants that are used, for example, extracting available phosphorus or potassium using Olsen extraction, a modified Kelowna, or ammonium acetate, they are indexes rather than being quantitative. And then we have the resins, the PRS probes. They're different again. They provide an indication of supply rate of nutrient, essentially a flux. That is the amount absorbed per unit surface area per unit time. So extractions then give the concentration or parts per million, weight per unit weight of available nutrient in the soil. And there's a lot of different extraction methods out there that are used to assess available nutrient in soil samples, folks. And I borrow this table here from the review on management of phosphorus fertilizer. Cindy Grant and Don Flayton depicts the different extraction methods used to assess soil available P in the northern Great Plains. There's a lot of different ones there. In fact, I had a student do a review of this, and going through the literature a few years back, I think there was at least 50 worldwide, but there's a lot of different ones as depicted there. And some of those work better than others depending on the soil conditions and the soil chemistry. For phosphorus, which kinds of phosphorus precipitates, calcium versus aluminum, for example, dominate, and that's often a function of soil pH. And this is an example then of taking those extraction values then and putting together a table that ranks those levels from extraction according to very low, medium, high, very high. And that's a very broad brush that you see here. And I'm not even sure where this table came from. I have it in my notes, but it's very broad brush. And as you can imagine, you know, it kind of gives you a general indication at first glance, but it's really not specific to the crop, to a yield potential, to a soil type. So it's a first brush. And then that type of information, though, in the actual recommendation system, and this is an example here of a rate recommendations for barley. So for a specific crop, this from AgVice Labs, for barley, for different soil test levels, different yields, and also for different placements. As you can see there, banding versus broadcasting, that's a sufficiency, and also providing recommendations there for application for sufficiency and also for maintenance as well. So tailoring those basic, is it, you know, low, medium, high, very low, sufficient, into an actual more precise, specific, crop specific, placement specific, fertility management strategy specific type of application. And just to show you some software that is available to the public out there in relation to this, one is the Alberta Farm Fertilizer Information Recommendation Manager, or a FIRM as the acronym. And this is a program that you can take the actual soil test results, the analytical results, and put them into this, and using curves and economic analysis, come up with a recommendation. For the longer term fertility management, maintenance approach, we have now the Prairie Nutrient Removal online calculator, a very handy, easy to use tool to determine what is being removed in the harvested seed. And then if you're interested in forages and straw and nutrient value removal in straw, the Ministry of Agriculture also has a nutrient value calculator for that, a simple Excel program. And I know folks in my 312 class have seen these tools. and that's the prairie nutrient removal calculator there. Many of you have seen that, a very easy to use tool to very rapidly determine for a yield what the nutrient removal is in the harvested grain. The other approach that's used here on the prairies to assess available nutrient is the use of ion exchange resin, which gives a supply rate of available nutrient. So it's a kind of dynamic measurement of release rate over time. And these ion exchange resins in membrane form offer the advantage of having a measurable surface area. So you can actually determine a flux or a rate of supply per unit area. They're pretty easy to use as well, and back in the old days, these were used in a kind of extraction, put in with soil along with water in a suspension, and this then really evolved then into actually encapsulating the membranes into a probe, and then for that being used in the lab or field, which we term the plant root simulator, and did lots and lots of work on this technology in my research program. And from a practical routine soil analysis, nutrient assessment standpoint, it's used inserted into samples of soil in the laboratory, Western Ag Labs, in samples of soil that have been pre-wetted. And so there's two types of probes in this that are used. The anion probe that contains a membrane positively charged that absorbs negatively charged ions like nitrate, phosphate, and sulfate. And then the cation probe, which has a negatively charged surface, absorbs positively charged ions, cations like potassium, calcium, ammonium, and other cations, micros, for example, as well. And so that in the case of this gets put into a program, the crop caster, which takes those supply rates, and as you can see on there that are measured, converts those using a model into supply rates in pounds per acre. Those are the red lines, kind of like a gas gauge, and that supply in relation to the root, the crop nutrient demand, with inputted by available water, including stored soil moisture, precipitation, the density, pH, EC that come from additional measurements made on that sample to come up with a recommended amount there that you see in the white boxes over on the right-hand side. All of these methodologies, regard whether you're talking about extractions or whether you're talking about PRS probes, require analysis of ions in the extracting solution or the eluting solution used to strip the nutrients off the membrane. So that involves instrumentation like automated colorimetry, wet chemistry, and also plasma emission spectroscopy as well, a plasma ICP that have in our research lab here in the department, shown there on the lower right-hand corner. So I guess sometimes question comes up, wrong soil test or soil test done wrong. Sometimes it's an inappropriate soil extraction for the region, and I'll talk a little bit more about that later on. But when it comes to soil tests done wrong, there's actually, from my experience, relatively little room for any mistakes having been made in the analytical lab in terms of the methods and the chemistry and the instrumentation that they're using, because those labs have strict quality control, quality assurance measurements or measures that they need to take. For example, running standard samples of known values that are in the analysis, that's done to assess the accuracy. Also replicate of repeated samples to determine the repeatability or precision, account for instrument drift. So I found when I've sent samples away in terms of the value that comes back for the same methodology, extraction method, actually really quite good agreement amongst laboratories. When it comes to assessing those soil test methods, that test method really should measure all or a consistent portion of that plant available pool from the soil. And many of these extractants are geographically limited, and they only work well in some soil types with specific chemistry. And I use the example here for phosphorus, where the sodium bicarbonate extraction, or Olsen P, tends to be favored for neutral to calcareous soils. It will tend to overestimate phosphorus in acid soils. We have the malic and the bray. Those are extractants that are designed, particularly the bray, for acid soils in particular, because those extractants like malic and bray work work best in soils where we have the phosphorus chemistry dominated by iron and aluminum, and it's those kind of compounds as precipitate or adsorb P that dominate. The other one is modified Kelowna, and that's one that tends to work well over a fairly wide pH range around neutrality, moving into slightly acidic and basic pH conditions. And for that reason, the results that you're looking at from a soil test for a method really should be one that's appropriate for your area. And some of the labs do provide you with values for a couple of those different extractions for example, for phosphorus. I think sometimes that the soil test analytical method gets blamed for an unreliable assessment, a poor recommendation, when in fact the high spatial variability, inadequate sampling to account for that variability out there is in fact the culprit, folks. And just as an example here, sulfur, I always call it the most variable macronutrient. Just as an example here, in one field, you got 5 ppm of sulfate on the upslopes throughout most of the field, but you got a saline area there with sulfate salts, you got 5,000 parts per million of sulfate sulfur. So you can see there in a kind of a traditional approach to sampling, the traditional where you just go across that field and you end up getting into that saline area with all that sulfate, you produce a composite sample that comes out of there. It tests really high in available sulfur. Yet a large portion of that field could indeed be and would be, according to the extractable, a sulfur deficient. So we really need to think about again about how good our sample is, and going back to the basics and thinking about how is that sample taken, how representative is it. One thing I want to make people aware of and to pay attention to when it comes to interpretation is note the units in which the nutrient levels in the soil are reported. These can be different depending on the lab. Some labs will report the soil test nutrient levels as parts per million. And parts per million is weight of nutrient per unit weight of soil. Micrograms of nutrient per gram of soil, or milligrams of nutrient per kilogram of soil. Other labs, some of them, may report the nutrient levels as pounds per acre. And in that case, what they've done is they've taken the parts per million and multiplied it by 2 to come up with pounds per acre or kilograms per hectare. And that applies for a 6-inch or 15-centimeter sampling depth, folks. And that conversion factor is based on longhand calculations, assumes a bulk density of 1.3 grams per cubic centimeter. So once you've got the analysis, then I guess the next step in this whole process comes into the prediction of nutrient demand, crop response to added nutrient. And folks, this is really, I think, the most challenging aspect. The soil test value, of course, is only one consideration. Ultimately, what is needed for additional nutrient greatly influenced by weather conditions, which we all know are very difficult, challenging to predict here in the prairies. We need to think about the crop, the type of crop, what's its yield potential, what's its nutrient demand going to be. We need to think about what's happening in that soil over time, mineralization, which may be accounted for example, through some measurement of organic matter, or it may be assessed directly, such as in the PRS assessment, solubilization, losses, fertilizer. We need to think about form, timing, placement. Those are all going to affect the efficiency and what those roots actually see. Presentation I was at just this last month, Dr. Don Flayton presentation, predicting crop response to fertilization. Not as simple as some people think, indeed. And Don made a really good presentation on outlining just all of the different things that are involved there. You're dealing with soil, crop, weather, field, economic variability. Challenging indeed. So we need good information. And a couple of sources of information or information that is used in recommendations that are made that come out from agencies making recommendations, benchmark field trials and models. And I guess it all comes back to the fact that a soil test value really is just a number. It's just a number until expertise is applied to turn it into an appraisal, a recommendation. And I had also the opportunity just this past month to see a presentation by Elliot Hildebrand on the expectation of soil sampling, which really depicts one of his slides very nicely. You know, we hope, and this is an example of 30 canola fields in northwestern Saskatchewan with similar rainfall. What we really hope is that, you know, as that soil supply of N increases, as shown there depicted, that the applied nitrogen, that is the nitrogen that is applied under those similar conditions, decreases, and that overall what we hope is that we see a relatively high and stable yield that indicates that the system is functioning properly. A little bit about benchmark field trials, and going back into history, a long way back. Once upon a time, there was a large publicly accessible database summary of what were termed expected yield increases from fertilization, and this came from many different fertilizer rate response trials that were conducted 60s, 70s, and 1980s. And in fact, this information collated together was the basis for recommendations that were made by the Saskatchewan Soil Testing Lab, the university lab back in the 70s and 1980s. Back then, things were fairly simple, though. Keep in mind, in terms of cropping systems relative to what we had now, back then it was cereal fallow, cereal on stubble, maybe a few other crops. We recognize that field trials, you know, and technology advancements that will come from new crops being introduced, new crop varieties, new rotations, technology advancements in fertilizer form, timing, placement, obviously will tend to date those benchmark databases. So pretty hard to go back into trials that were run in the 1980s and really have them have a high degree of relevance to what's going on in most of the prairie cropping systems. And I think the complexity of today's cropping systems also makes the collation of all that together challenging. But those types of yield response trials still serve a lot of value. The information is disseminated and invariably gets built into the recommendation models that we see being used out there in industry today. Nitrogen fertilizer response curves. I show here just that concept that may come from a benchmark trial, adding different rates of fertilizer and the yield increase documented under different soil nutrient availability conditions as a benchmark type of trial. We're hoping there, folks, that everything that was experienced in that trial is going to be, you hope that your growers' conditions are going to be as similar as possible to that. So important to have a really solid database that covers all the different conditions that exist out there. And certainly those benchmarks can reflect local conditions well. They're easy to apply economics to, but they are expensive. Field trials are not cheap, and they can go out of date fast. And as a consequence of that, we still see benchmarks used a fair bit for nutrients like phosphorus and micronutrients, for example. But for nitrogen, a lot of the systems now, instead of using benchmarks, use a model, a nitrogen balance. In this case here, for nitrogen fertilizer required is determined in this balance by the nitrogen demand by the crop, subtracting the nitrogen that's coming from the soil. It may be a measured mineral N, taking out what's coming from mineralization, which may be estimated, or it may be actually a component of it assessed or included, modeled out, and also considering the effect of the previous crop in some cases, residues on immobilization, mineralization in the model, and also nitrogen losses. So a lot of different things then that are built into there. In some cases, may be measured directly. The nitrogen in the soil as mineral N, also a component of the mineralization, for example, in the PRS, and then other elements of the model that are used to provide a model output, the additional nutrient. Models are easy to update because you only need to update one component of the model rather than go back and run the whole trial again. It is a little bit more difficult to apply economics. In the case of the PRS nutrient cropcaster. It's a mechanistic model based on Dr. Stan Barber's principles of nutrient bioavailability, diffusion, movement of nutrients, and uptake by roots to develop the supply rates and the crop root demand, as shown over here. The model also does incorporate some response curves here that can be toggled back and forth, and also used to determine the optimum economic rate. Just to make the picture complete, there's soil health tests out there that measure a number of different biological attributes as well as physical attributes, things like aggregate stability, density, but biological attributes specifically like microbial respiration, diversity, enzyme activity. We measure lots of these in our research work, looking at the influence of different management practices. Those types of measurements do have some relevance to soil fertility, nutrient availability. Typically, those kinds of soil health measurements, though, are not used for specific rate recommendations, but still can be helpful in making a general assessment of biological activity in the soil, biological processes that affect nutrient availability, like mineralization. I think to be most useful, any type of soil assessment does need to be calibrated and/or incorporated into a model, and also validated in different environments. I guess I'd say common scenario that comes up, and I encounter this many times. I send the same sample of soil to 3 different agencies and get 3 different recommendations. What's up? Well, I guess I would be surprised if you got the same recommendations out of 3 different agencies. I would expect them to be different because of different approaches that are used in the analysis, the prediction model, the economics, the kind of risk adjustments that may be made. And I guess I always think the correct fertilizer recommendation really is determined in hindsight. And that's why I think it's very important to follow up, to verify that soil assessment, that prediction model performance, that model validation, so that in fact that model can get better over time. And of course, sources of error, garbage in, garbage out, very much applies. And it goes sometimes all the way back to improper sampling and sample handling. Maybe it can be something as simple as failure to provide the correct information. Things like legal location. In some models and some systems, that's very important in establishing what soil climatic zone the grower's in. So if you're west of 2nd versus west of 3rd, that can make a big difference and put you in the wrong soil climatic zone compared relative to where the field is actually located. Soil water storage, very important, and it's important in all models here, all recommendations. I think what we see here on the prairies, because in our semi-arid water limited system, that soil water storage makes a very important contribution to the overall all water availability and yield potential of that crop. Cropping history, previous crop yield, residue management, those are all important. Another thing I would mention that I see as sometimes an error is folks want to subtract the nutrient levels in the soil from the recommended rates. All the systems that are out there, in the recommendation that comes out, that available nutrient level in the soil is already built into it. You don't need to take the nutrient level in the soil off of the recommended rate. And also perhaps selection of inappropriate yield goals, maybe too high in some instances, but also too low in some instances. And that's where models, I think, that are sensitive to all of the factors that affect yield on a grower's farm are very useful. So the last thing I want to talk a little bit about is information that's found in a typical soil test report. And I have here as one example a soil test report. This was one from U of S Goodale Farm back in 2017, pre-COVID, from one of our sampling trips out. And I use this. ALS doesn't make nutrient recommendations anymore. They still do soil analysis, but I use this just to show kind of the general components that you'll find in any kind of a soil test report, like this here on a piece of paper here, showing some information here that's provided, the legal location, which allows the lab to position it in a soil climatic zone, some other information that's useful. You've got the soil test characteristics here, which I'm going to talk a little bit about later on. Your soil test nutrient levels, another section here for the sample of depth that was submitted, in this case 0 to 6 inches. Oftentimes you'll see a general rating of deficient, marginal, sufficient, excessive, a bar graph like that. And you'll note here, in this case, the nutrients that are used to express the units that are used to express the available nutrients are pounds per acre in this case. And then you've got your nutrient recommendation rates down here on the bottom with some different recommendations based on different target yields according to probabilities of precipitation. And finally, at the bottom, some recommendations and comments, additional ones that I think are always useful to pay attention to that help in the interpretation. Another report here, this is one from one of our research sites in southeastern Saskatchewan. Same kind of concept in terms of the structure of the report. Available nutrients, you've got some bar graphs here, you've got the recommendations for different target yields and some comments at the bottom here. But one of the things I point out here in this case, the levels are expressed as parts per million. So if you want to compare these levels, make a valid comparison then to the other analysis there, you'll need to multiply those by 2 to get into the same units needed for comparison, or take the other one there and divide by 2 and back into ppm. And I stress this because this is sometimes a common source of confusion. And so pay attention to the units, and if you want to compare, make sure that you do the conversion so that you're comparing apples to apples in terms of units used in the expression of the nutrients. Kinds of things that affect soil test nutrient levels over time: the past crop yield, nutrient uptake, removal. Also, we need to be thinking about things that might occur between the time you took that sample and got it analyzed in the fall until the spring of the year, things like weed, volunteer growth, maybe some additional losses that may have occurred, especially important for mobile, easily lost nutrients like nitrate. There can be significant changes depending on the conditions from when you took that sample in the fall till the time of seeding and when that crop starts to take that nutrient up. So things like that, fall weed, volunteer flooding, and it's, I think, always a good idea to go out and maybe check a few fields just to see how much of things change between the fall and the spring. Okay, lastly, other properties that are commonly assessed in routine analysis of agricultural soil samples. I just want to say a few words about pH. And one thing to keep in mind is for pH, the measurement method will affect the value. pH that's measured in a calcium chloride suspension will be about a half a unit lower than in a water suspension. So again, some labs may use, a few labs may use calcium chloride. Most use a soil water suspension, but again, be aware there could be a difference. Generally, we find pH increases with depth. Electrical conductivity. Another value that a property that's commonly measured in a soil test and used to provide a salinity rating. And sometimes that salinity is linked to yield in models where if the soil is saline, there'll be a reduction in yield potential and correspondingly nutrient demand, and that's reflected in the recommended rate of nutrient. And as I mentioned earlier, EC, salinity, closely tied to sulfate content in our prairie soils because it's generally sulfate salts that cause salinity. So even in a soil that's rated according to the EC as only maybe slightly saline, typically you'll see oodles and oodles of sulfate, and generally EC increases with depth. Organic matter, measured by a combustion analyzer nowadays, used to be Walkley Black. What they actually measure is carbon concentration, then multiplied by a factor which gives you percent organic matter. That may be linked to mineralization prediction in some models, and again, it decreases with depth, typically with your incremental depth soil samples. The last one, cation exchange capacity, composition of exchangeable cations, percent base saturation. Cation exchange capacity, or CEC, very much influenced by texture and organic matter. So if you got a soil that's high in clay, high in organic matter, that means high cation exchange capacity. And the CEC is a measure of the net negative charge of the soil. And so that tells us about its ability to hold cations, nutrient cations, base cations like calcium, magnesium, potassium, sodium, as well as ammonium, and also acid cations, acidity, like aluminum and protons. I look at the composition of exchangeable cations on a soil test. One of the things I'm often most interested in is the sodium. A high percentage of sodium relative to cations, for example, greater than 15% sodium on the exchange complex, indicates a sodic soil. A soil with issues probably with a hard pan, dispersion, and your exchangeable bases then, percent base saturation, calcium, magnesium, potassium, sodium as a percentage of CEC gives base saturation. Oftentimes we see soils here in Saskatchewan close to 100% base saturation. I don't use this much here, more so probably in more highly weathered acidic soils. And all of these properties are useful in monitoring over time, coming back to the same location, following, for example, pH, organic matter content. And I put this up here just to show some pH values from a study that I'm involved with, Prairie Soil Carbon Balance Project with Ryan Hangs and folks from Ag Canada, Brian McConkey and Mervyn St. Luce. Soils that were taken, samples from across Saskatchewan farm fields in 1996 and sampled again in 2018 in exactly the same place, and showing the trend in pH there. A little bit lower pH in the as we move, pH slightly decreasing from the brown, dark brown, black, dark gray, and gray, reflecting increased weathering. Over there, really not showing much difference in pH from 1996 to 2018. But these were also soils that had been under conservation management and actually showed increases in organic matter content. And we know that organic matter content can help a buffer against pH change. But we're certainly becoming aware there are a few locations and areas within fields where we are seeing some low pHs coming out. Monitoring things like pH in those exact same location over time, soil testing, a very effective tool. So some final thoughts. I think a properly collected soil sample, folks, can tell you a great deal about fertility and conditions for plant growth. Great foundation for predicting the need for additional nutrient, identifying soil properties as the basis for a fertilizer recommendation that is site specific. Very much an important part of our move towards a site specific precision application. Enables us to take a look at how our management practices are influencing soil productivity, health over time, and certainly overall, I think, becoming more important in this new age of documentation. So with that, I think I've used my 45 minutes, and I will stop right there, Erica. And thank everyone for their attention, and now we have questions.
Thank you, Jeff, for that great presentation. So we'll now take any questions that you may have for Jeff. And so we do have a few queued up, ready to go.
Okay, I'm just going to get my oh, there I am, I'm on. Yeah, and I will just shut my, actually I maybe leave my presentation up in case I need to go back to it. Yep.
Perfect.
And a drink.
Is there any correlation between actual nutrients applied at seeding and their availability and the water fraction volume in soil at different depth?
So the correlation between nutrients applied at seeding and what you would find in the soil, in the case of nitrogen, and being able to find that nitrogen in the soil test, of course, how much you find in the end is going to depend on how much you might be losing upon application. Even, for example, for anhydrous ammonia, the ammonium form of nitrogen, if you're just measuring nitrate in the soil test, you won't see that ammonium nitrogen in the soil test extraction. If you're just measuring nitrate, you will see the supply in a PRS measurement because it measures ammonium and nitrate. So you should be able to see that nitrogen in the soil test. In the case of phosphorus and potassium, depending on how much you're adding, those pools that are being, in the case of an extracting solution, those pools that you're measuring are pretty highly buffered by other fractions. So you may not see a huge increase in those in the soil, but you should generally tend to see some increase in the level. And I guess as time proceeds and that nitrogen, the ammonium, converts to nitrate, that nitrate could indeed move down through the profile with water, and then you may see a difference in distribution. So I think maybe that's what that person was perhaps looking for.
Wonderful. Thank you, Jeff. So the next question I have, it's seen claimed that PRS probes better reflect actual field conditions and nutrient availability than traditional soil sampling and chemical extraction testing. How true is this claim, and should more farmers be looking at this method?
Okay, so I guess, and I'm going to come at this with a little bit of bias because I was involved in the development of that technology, and really to take that and actually insert those probes into the field or a sample of soil. And what we saw in our early work was, in some cases, better correlation with plant uptake, with a supply rate compared to an extractable amount that was related to, we felt, a couple of factors that we established. One is that you are documenting or including in that assessment how the physical condition of the soil, the structure, and the texture influences the ability of the nutrient ion to move from where it's located to that surface and be absorbed, similar to what a root sees out there in the field. And the other is that it also encompasses, since it's a flux, a supply rate over time, essentially accounts for some of the release rate or the ability of nutrient ion to move off of the solid phase or be released from decomposition into available forms and be taken up by the plant. And so that's really why that technique really, I think, that approach, I found it very appealing and very mechanistically oriented and following along the lines of really what I'd learned a lot about factors affecting nutrient availability from my time, particularly, especially in graduate studies.
Wonderful. Thank you, Jeff. So the next question we have is, do you know why the prairie nutrient removal calculator didn't also include uptake values for crops? As an agronomist, I've been looking at uptake values compared to removal.
And I think I can comment on that. It has to do with a lack of data on straw. So the grain is an easy thing to, I mean, it's an easy thing to get, and even in research work, and you go into the past literature, there's lots and lots of studies where they harvest the grain off the plots and they measure the nutrient content in the grain, a lot less in which they've measured the nutrient content in the straw. And of course, it's the straw plus the grain that represents the total nutrient uptake. And there's probably even less studies that have measured how much nutrients in the roots, because those are real. That's a real struggle to measure nutrients in roots. So that's why you don't see as much on total uptake as you do on grain removal. And the way that my understanding in the prairie crop removal, in collecting that database from actual farm fields, was a whole wide range, large number of samples, was to deal with the grain only, because that straw is a whole lot harder to get. Now, the straw component and the forage removal, again, that as what I use in that is the SMA calculator, that Excel calculator, and it's got yields for values for straw removal for forage crops.
Wonderful. Thank you so much for your answer, Jeff. So the next question we have is, if you're used to reading a soil sample that uses one extraction method, say Olson,
Yep.
What is your advice on how to interpret another soil sample test result that uses another method?
Yeah, very good question. And there are correlations and equations that can be used to convert from one to the other. I know I've got some publications that I've been involved with that provide that. I know also, I think John Hurd in Manitoba, he has some information on that as well, a poster, and they might even have that online somewhere. But it is possible, if you do a search, to be able to go from an Olson test to a modified Kelowna to a Malik. You can do that. But remember, they are correlations. So there's a general equation that generally fits the data, but there's a lot of scatter around that. So again, I think you're probably best to have the actual assessment done using the appropriate method rather than correlating from one to the other. Because the R-squareds are not 1. They're typically around maybe 0.8. So they're not perfect relationships. but it'll get you there partly. It'll get you a good ways there.
Wonderful. Thank you, Jeff. We just have a few more questions for the moment here. So how do you convert from parts per million to pounds per acre if the sample depth is 0 to 12 instead of 0 to 6?
Easy question. Multiply by 4. Yeah.
Wonderful. Thank you, Jeff. Now, does the electrical conductivity reading taken before seeding give any idea on the approximate nitrate nitrogen pounds per acre available or not?
Typically in our soils, the amount of nitrate as an ion compared to the other ions like sulfate is very small. So the EC is really reflecting calcium, sodium, magnesium, sulfate salts, maybe chloride in some cases. The nitrate would be a very limited contributor unless you were dumping fertilizer, like dumping it out of your fertilizer box out into the field. So for that reason, I would expect your electrical conductivity measured on that sample to have very little relationship to the nitrate that's contained. But on the other hand, I will say this: saline areas within a field where you've been running your fertilizer applicator across there over, and you've been putting on fertilizer nitrogen and have had very little or no crop utilization of that nitrogen, you will see oftentimes high nitrate levels there because the crop hasn't used it. So there could be some correlation there, but yeah, no, the EC that you measure in that sample in a typical well-drained soil, it's not going to be a very good indicator of nitrate.
Excellent. Thanks so much, Jeff. So this is our last question for now. So in what conditions will you find surface pH higher than subsurface pH?
Okay, good question. And I mean, I guess one example that I can think of is soils where you have a AE horizon. You may have an AP. Right at the surface, where you've had some recycling of bases from depth by your crop or your forage that you're growing there, deep rooted, taking calcium out, the bases up, putting it on the surface. But below that AH or AP horizon, you've got a highly leached AE horizon. And that AE horizon may be an area where you've had clays alluviated out of there, other cations, and would be of lower pH. You also might expect to see a lower pH, perhaps down a little bit deeper below the surface at the depth where you're putting your nitrogen fertilizer down every year, because your nitrogen fertilizer, ammonium, converts to nitrate and it produces protons or acidity. So that can also be responsible for a zone of lower pH in the soil below the surface.
Excellent. Thank you very much, Jeff. And we will not be able to get to all of the questions today, but you can send them directly to Jeff afterwards. So thank you very much, Jeff.
Thank you, Erica.
So next up, we have Derek's presentation. So whenever you're ready, Derek.
And let me show. Okay, can you see me and hear me and all that good stuff now?
Yes, we can.
All right, perfectly. I'm gonna hide my webcam so I can see my screen. All right, here we go. So today I'm gonna, first of all, thanks everyone for the opportunity to come and share some of the research that we've done, and also thank Jeff for the talk that he just gave, because I think in a lot of ways it sets up some of the stuff that I'm going to talk about today. And I'm going to talk sort of about soil analysis and soil chemistry research that you can do by applying spectroscopic tools to soil samples and to agricultural fields directly. And really the driver of this is how complicated that soils are as a substrate for looking at chemistry. You know, if you ever play 20 questions when you're on a car trip and you choose soil, then when the person asks animal, vegetable, or mineral, then the answer is yes, right? Like they're an extremely complicated mixture of solids, liquids, and gases. They're crystalline minerals and poorly crystalline minerals and organic materials. They're full of living things that are undergoing metabolic processes, and they're dynamic samples. They're alive, which make them really hard to measure and analyze and characterize. And on top of that, they're going to be affected by the weather, affected by the climate, affected by your management strategies and what crops you grow on them. And as Jeff mentioned, they have variability over all spatial scales, but from a chemistry standpoint, from the atomic scale all the way to the landscape. And when you put all of this stuff together, then that soil sample that you put in a box and ship off for fertility analysis is quite complex, but it's also just a snapshot of what was going on in that soil at that moment that you collected the sample. And snapshots are useful, a picture's worth 1,000 words, but when you're looking at dynamic processes, for example, this gymnast on the left, it would be really difficult to assign a score to this gymnastic performance from any individual snapshot, right? Like the dynamics, the process, the overall flow is what really matters. The same thing is true for soils, where looking at processes like soil carbon, we understand now that it's not just soil organic matter is not just a recalcitrant, stable form that's going to persist over long term and we can sequester. No, it's actually a complicated biogeochemical process where there's constant inputs, there's constant metabolism of those plant and inputs, and there's adsorption and aggregation, and there's microbial processes that are eating it. I apologize, my cat is climbing on me right now. She ignored me for about a week, and as soon as I give a talk, she's jumping on me. But that combine to have a residence time or a turnover. And so if we want to manage the carbon in the soil, we have to understand these processes, and it's very dynamic. All right. What we learned from that in summary is that looking at the form of nutrients, the form of carbon, the form of minerals that are in our soil is often a lot more important to describing the system than the total amount that's there. And you know this already, and Jeff talked about this already, that the total amount of a nutrient might not be really all that relevant. It's what fraction is plant available. So we already do speciation. It's just that we're doing it based on what the plant can access. And so then, what is a soil sample? It's kind of a philosophical question. But that situation out in the field where a soil is a mixture of about 30% or 35% water, when we take it into the lab to do characterization or do analysis, then we've lost that really important phase, right? And so I would say from a soil chemistry standpoint, from a spectroscopy standpoint, one of the most important decisions that you make is whether you are going to dry your sample and grind it up and be able to consolidate it like that, or if you're going to try to look at it as it is or as it exists out in the field. Now, right away you would say, oh, I want to look at it as it sits. I want to understand it as a whole soil. But drying it and grinding it is going to give us the ability to have much better replication. It's going to give us a lot more inference space on that sample, and it's also going to really expand the types of analysis that we can do. You're not going to be able to do combustion for organic carbon on a wet sample. You're not going to be able to do a lot of different kinds of spectroscopy on wet samples either. So operationally, we've changed our samples to be able to work with them in the lab. Now, having said that, that I've spent however much time, 5 minutes now, talking about how hard this situation is. But soil testing is thought of as kind of a solved system that's pretty easy. And you know, I pulled this off of the web, and it is easy as long as you exclude the cost, the time, the effort, all of the waste in terms of both taking your sample, shipping it, and then when it gets to the lab, there's chemical waste that's produced. A lot of times these are acids and reagents and analysis waste. But then on top of that, your soil, which is a valuable resource out in the field, has to be disposed of as waste at that lab, right? And then results have to be developed. Jeff talked about modeling, and then how robust or how reliable they are really depends on how many samples that you were able to afford to ship off, and then also how they were collected in terms of timing, and then also the robustness of the measurement. And so this is a situation where it's easy in principle, but in practice, it often is not able to really get to the heart of what we're interested in. And so then, you know, as a chemist, I've turned to spectroscopy. And so I wanted to talk a little bit about how it can help. And before I do that, I mean, I guess, so I'm from Louisiana originally, and I moved here because Canada's only synchrotron, the Canadian light source, is on the University of Saskatchewan campus. And synchrotron science is really powerful, and it's the primary tool that I use. I'm not going to talk about it a lot today, but what it allows us to do is look at the chemical form of nutrients or contaminants that are in soils at very great detail, and then look at how they're affected by landscape position, by how they're affected by management strategy, and how they're changing over time. So we can look at the chemistry of a sample at the atomic scale and then understand what's happening out in the field. But what that also means is that we're starting off with big field scale research projects, and then we're kind of condensing them down to a few samples that we can look at with a very advanced technique. And so we're using our intuition as agronomists, we're using our intuition as ecologists to choose samples to look at things where we think there would be good contrast, and then we're trying to understand why they're differing. And then from that, understanding how the chemical form relates to the nutrient availability or the nutrient status or the performance of that site, then we go back to the field, we come up with a simpler test, or we come up with a simpler treatment, and then we kind of iterate until we've solved that problem. And so, yeah, so historically, we use spectroscopy to improve our lab-based soil tests, or to choose what soil test is most appropriate. And that's kind of different than how people think of using spectroscopy in ag, and what I'm going to talk about for most of the rest of the lecture. And so then, before I really get moving on that, you know, why do we care about fertility analysis anyway? You know, it's probably a reasonable question. And if you go back to some of the underpinning concepts of soil fertility, it's really the idea that the yield response of a cropping system is going to be limited by whatever nutrient is most deficient. And so if we can identify that and alleviate that deficiency, then some other nutrient will be deficient. And if we can satisfy by adding all of the fertilizer nutrients so that they're no longer deficient, then we're going to be limited by things that are out of our control, right? Like the light, which is going to be primarily a function of latitude and moisture. And at that point, we've kind of optimized the system. So we want to add our fertilizer such that we are getting the optimum crop yield in the least amount of money in the shortest amount of time without over applying it. And so that's kind of the basis of soil testing and soil sampling and soil fertility, really, when you think about it, is where are you at now versus where you could be. And that where you could be is you know, built the yield response curves for different nutrients is really where you get that. And you know, to the right here, when you're looking at a soil test phosphorus level, then what you see is when your soil test levels are low, then there's a very high likelihood that you'll get a response to yield if you add more. And then when you have reached a sufficient level of fertilization, then you're not going to get that increase anymore because that nutrient's no longer limiting yield. And so then what soil testing and what Jeff spent a lot of time talking about was choosing an extraction, which is really trying to simulate or estimate what that fraction of the total nutrient in the soil is that's available to your plant. And so this was historically built on experimental trials where you would add variable rates of fertilizer for a particular crop on different soil zones, different soil types, different varieties, and get a relationship between how much phosphorus is in the soil and how much extractant how much was phosphorus is in your extractant, and then what that yield was. And so then, a lot of our fertility recommendations, a lot of our soil test values are built around this concept. And so, spectroscopy works a little bit differently, where we're trying to fractionate the phosphorus in the system based on its chemical form. So what fraction is adsorbed, what fraction is organic, what fraction is calcium phosphate, what fraction is aluminum phosphate. We directly measure those in the solid state. If you have a spectroscopic tool that's capable of seeing the phosphorus. And so then that gives you sort of a highlight of why you might want to use spectroscopy, right? You don't have to do those chemical extractions. You could directly peer at the chemical form in the solid soil sample itself. So this non-destructive method means you still have the soil sample afterwards if you want to collect a spectra and then run it through your lab analysis. Then you can calibrate your spectroscopy with your normal soil testing procedures. The measurement of an individual spectrum is usually quite inexpensive. Synchrotrons would be the exception to this because the facility costs a huge amount of money to build and run. And so synchrotron spectroscopy is not going to be a routine soil test method. However, we can use that really sophisticated tool to find a simpler way to improve soil tests, right? And what I would say about spectroscopy, and the reason that people are really focusing on it as a tool for agronomic soil testing, is that at the worst, you could insert a spectroscopic instrument, and we'll talk about near IR in a second, but whether it's near IR or mid IR or X-ray fluorescence or some kind of spectroscopic tool, you can put it in your soil testing lab and replace all of the cost and the expense of doing extractions and digestions for soil test quantification. And that in itself is pretty good because it costs 5 or 10 times less per sample once you've invested in the equipment. And so right away, you could measure more soil samples, which is going to give you a little more confidence that you've captured the variability of your field. But where the real power of spectroscopy is the idea that you could take these instruments into the field and directly measure spectra of the soils without having to dig them up and ship them to a testing lab. And spectra are just how light interacts with materials, is also then really compatible with remote sensing from satellites, with hyperspectral applications from drones. And so a lot of our digital ag and a lot of our big data applications, we could add a soil fertility component to. And then another couple of things that are maybe not quite the first things you think about when you think about soil spectroscopy is if you could build an inexpensive sensor that could measure a particular soil property, you could look at leaving them in the field, continuously scanning, and then just uploading them to the cloud, so that instead of once a season or once a year or once every few years measuring what's going on in your soil, you could dynamically measure, continuously measure many times a day. And having that quantity of data then sort of feeds a lot of machine learning and AI models that you maybe not able to use now with soil testing. And another thing that I think is potentially really important is if you're collecting spectra of your sample of your soils and they're GPS located and you've stored them digitally, then you can go back and you can mine them later as we start to understand the spectroscopy better. And so in some ways then every field, every position in your field even is potentially part of a long-term agricultural trial. And you know, I think that's kind of underappreciated, but once we start collecting this data, I think it will really provide a lot of new information to agricultural science. All right, so then let's look at a spectra of a typical soil spectroscopy project right now. Involves near infrared from about 2,500 nanometers to about, I guess about 600 nanometers or so. And near infrared is a rapid, non-destructive way, as I mentioned, to measure soil properties. It's really sensitive to how much water and how much soil moisture is in a sample, to organic carbon content, to iron oxide and chromophore content, and also to texture and clays. Research that uses near IR spectroscopy usually collects a large number of soil properties. So your soil test outputs, you know, CEC, pH, plant available nutrients, organic carbon, texture, and so forth and so on, and also collects spectra, and then just uses correlations to look at what properties of the soil are good at, or in near IR is good at measuring or monitoring. And the nice thing about this is you can correlate, or you can come up with coefficients for a lot of different properties out of that single soil spectrum. And here's a paper that I pulled off the internet from a few years ago that kind of looked at comparing the performance of spectroscopy, which is the soil attributes analyzed by the sensor, to the performance of wet chemical techniques in the lab. And for some analysis techniques, you can see the closer we are to the outside of the circle, the higher the R squared, or the better the performance of the technique. And so what you could see out of this is kind of an estimate of what soil spectroscopy could be good for. And you can see it does an okay job of looking at organic matter. A very variable performance in terms of phosphorus and plant exchangeable base cations. CEC again, pretty variable, and sand, silt, and clay again kind of variable, depending on what the instrument that you're using and how the samples were prepared. You get a lot of variability in the spectroscopy. In the lab, you get better agreement from lab to lab, but if you will look at how close we are to the center of that circle, some of these techniques are still not great. But what I will point out here is a quote from this paper, which was that for plant available phosphorus, the R squared with using spectroscopy was never better than 0.36. And that's, you know, we're getting in coin flip territory now for estimating what the plant available phosphorus is. And the reason for this is that we're below the detection limits. You can't directly measure the amount of plant available phosphorus in a soil with near infrared spectroscopy, but instead you can see things that correlate to plant available phosphorus, like when you have more clay, when you have more iron oxides, when you have more organic matter. That's probably going to be a situation where there's also more plant available nutrients in a lot of systems. And so it's an indirect measure, which is not going to be as robust as things that you can directly see, like clays and organic matter. And that's kind of borne out when you look at sort of the literature about what near IR spectroscopy is good at. And what they find is organic carbon and total nitrogen. And organic nitrogen, it works very well. And that's over a big range of soils, over a big range of instruments. It's pretty good for that. It's also pretty good from a physical standpoint at looking at water content and moisture in soils, again, because water absorbs very strongly in the infrared, and it does an okay job at looking at texture and soil and particularly clay content. And so, that's working in the lab under conditions where the samples have been dried and ground, and already we run into some issues. But what if we just wanted to take them out in the field? Well, if you get a field portable unit that is going to last over an entire year of going out and sampling, they cost about $100,000. And if you try to use an inexpensive one, which is what we've done in our lab, because $100,000 per person going out in the field just isn't viable, then they break. So that's a problem. And so what we've found is that to go do these soil spectral measurements in a Saskatchewan field means that you're either going out and using a Dutch auger to get loose soil and probing them on the surface, or you're taking that auger hole and then inserting the probe into it as a function of depth. And both of those have some issues. How good the contact of the soil is with the instrument's window has a big effect on the performance. How the soil is aggregated, how much moisture there is in the soil, how much light is bleeding into your measurement from either from the being outside, or how good you're capping that hole if you're trying to plug it into the soil. All these have an effect on the measurement that you get, and also on the repeatability of the measurement, and also who's doing it seem to have a big effect. Now, having said that, we can measure about 50 samples a day by going out with a near IR probe and measuring them in the field by augering. This is slower than agronomic commercial lab soil analysis by just pressing hydraulic punches and shipping them off for analysis, right? Now, having said all that, those are mostly engineering problems that we could solve with buying a lot more equipment and revising it and improving it if the underlying spectroscopy is sound. So that brought us sort of at our research topic is this underlying technology sound? And the answer is kinda. You can take near IR scans of several hundred soils. You can process them with different algorithms. You could visually look at how they cluster, and total nitrogen, for example, you can already sort of see if we separate it into quintiles, that it's kind of following a visual trend. And then if you do partial least squares or some sort of quantitative analysis with whether it's machine learning or even simple models, you get a pretty decent correlation. I mean, these are just soil samples that we haven't done any extractions on, and we could get about a 0.9 R² for total nitrogen. And you can see our model is very close to that 1-to-1 line. So it kind of works in some systems. So then could we replace soil testing? Or could we supplement soil testing in a meaningful way? Well, one area where there's both great potential but also a lot of current problems is if we were able to collect spectra every few centimeters as we went all the way down to depth in the soil. I mean, that would automatically give us a lot more insights, a new sort of depth component to our soil testing. And we know that consolidating all the samples as a function of depth is giving us an average that may not always be good, but currently, as I mentioned, our rate of doing that sampling or spectroscopy out in the field was just so slow that it wasn't really going to be economically viable. So then going into the lab and trying to build a model with dried and ground samples is kind of where we moved. But we've also then took those samples and air dried them without grinding them, and also measured them at normal field moisture and structure conditions. And what we found is sort of the same thing that everybody else in the literature has. We can build a good model and we can calibrate things, and we can get pretty good performance on those ideal soils after we've dried them and ground them. But even with large data sets, as we start to have more realistic soil conditions, we have a difficulty in building those models. And so, I think there's still a lot of research to be done if we're going to try to do spectroscopy as the only soil sampling method or only soil analysis. And then what typically happens is people say, well, the reason it doesn't work is because you just don't have enough data yet. And if you had more data, then we could big machine learning our way through this. And so we collected what I think is a pretty good set of soil data from Saskatchewan. It's about 2,200 samples that were collected from academic and agronomic trials in a few different places. They range from 0.1 to 8% organic carbon, from below detection limit to 0.5% nitrogen. And we measured them all with the same lab techniques, and then we also measured them all with near IR. And what we found is we could certainly build a model that was reasonable. We found that doing continuous wavelength transforms gave us the best result in terms of pre-treatment. We found it didn't matter so much how we built the model if our data set was good. I'll show you some results here. For organic carbon, we found over those 2,000 samples, we could get about a 0.8 R squared. Not perfect. For total nitrogen, the numbers are much better. You can see nitrogen in the form of protein directly with near IR. It's not spread over the whole spectrum so much. It's in particular wavelengths, and so it performs. You can see we're closer to that one-to-one line, so the predictions are better. But at the end of the day, what we found is that it works okay for things that are, like organic carbon and total nitrogen. For things like plant available nutrients, the R squareds were really not useful. And the other thing that we learned is that it's not just purely about the number of samples. And in fact, increasing the number of samples doesn't always improve your model at all. What mattered was having a good coefficient of variation in your system. And what that means is that the variability of your sample data set needs to be kind of evenly distributed, or at least normally distributed, if you're going to improve the model by adding more samples. And the other thing that was interesting about this is the systems that really struggle where there's lower variability are areas that are more intensively cultivated. So, areas where the organic carbon doesn't vary so much because of management practices. Typically, the model had more struggles than in areas where academically there was a control, there were treatment effects and things like that. Then we had kind of built a system of soil samples that had a lot of kind of normalized variability, and the model worked better. And so I think that there's some hidden challenges in going from a concept to a commercial product in spectroscopy for agricultural samples in particular. And so to summarize, I think spectroscopy currently makes a lot more financial sense when you put it alongside soil testing than trying to go in the field directly and replace soil testing. Now, having said that, there are some parts of the world where estimating soil carbon is actually good enough to make fertilizer recommendations. It needs to be an area where increasing soil organic matter means that you can add more fertilizer and you get more yields. And in those systems, tropical agriculture, often what you find is that if there's not any organic carbon, it means there's not very much clay, and you're going to have moisture retention issues, and you also have more soil acidity problems. And so the organic carbon buffers acidity, the organic carbon preserves moisture in the soil, and so where there's more carbon, you can add more fertilizer and you get more yields. That's not a simple relationship in Saskatchewan agriculture, and so spectroscopy may not be a simple tool in this part of the world either. But what I will say in closing is that spectroscopy is a bunch of different tools, not just near IR, and there's a lot of different ways that we could better perform soil testing by understanding what's happening at the atomic or the molecular scale. And then, in closing, if we had more holistic things that we were wanting to look at, like soil health, or broader things like management zones, or how moisture is being affected by depth, those kind of short-term targets are probably better for near infrared spectroscopy and for field-based spectroscopy than plant available nutrients. And so with that, I will point out that I'm a professor, and so I just profess. I have students, they do, and also that some of this research came from a variety of sources. And so with that, I'll try to answer questions that people have.
Thank you for the awesome presentation, Derek, and we'll now take any questions you have for Derek. So I do have a few ready here. You touched on this a bit, but can soil sample results from spectroscopy analysis be compared to a wet lab sample, or is it not a fair comparison?
I think it depends. So, for synchrotron stuff, we can correlate the peak height of phosphorus with the total phosphorus in a sample really nicely. Like we can do fundamental measurements. For organic carbon, we actually get pretty good results. See, and the nice thing about spectroscopy is at the same time that you're collecting soil organic carbon, you're also getting inorganic carbon, and you're getting total nitrogen, and you're getting quite a few things. And so for those tools, it works pretty well. For ammonium or nitrogen or plant available, you're below detection limit, and I don't think they correlate very well at all.
Excellent. Thanks, Derek. So the next question we have is, if you used a portable unit on a field, would you take multiple readings from the soil surface and average out the data, or would you dig up a representative sample and read one sample? There seems to be many options available with this technology. What is the most accurate?
I think the real power is, well, I have funding and I'm an academic, and so I would say you'd want to do it all. You'd want to try to collect a lot of different points, and then also send those samples from different points to build a calibration model. The hope is that once that model is built and you've calibrated that system, for a soil zone or for Saskatchewan or for a region, then you wouldn't have to send every sample back to the lab for validation. That maybe 10% of your samples you sent for classical chemical analysis, but the rest of them you're just relying on the spectroscopy. And so it's one of those kind of feedback things where the more spectroscopy you do and the more samples you send out for calibration, the better the model is. I don't think spectroscopy in agriculture is ever going to be as good as wet chemical techniques for measuring a particular thing. But if you can measure 100 spectra for every one sample that you ship off, then you're going to get a lot better understanding of the variability of your soil or of your field.
Excellent. Thank you for that answer, Derek. So is there potential to use spectroscopy for plant tissue testing?
Yeah, it works much better for plant tissue testing than it does for soils. You lose a lot of the matrix effects, for sure. And I think the other thing I didn't really fully touch on it is with spectroscopy, you're just calibrating or correlating to anything. And so you actually could correlate or calibrate to your yields, and short circuit some of the chemical analysis testing stuff. But yeah, I mean, you should be able to link it to greenness or NDVI or NVDI, whatever. It's just a simple regression once you've collected the spectra.
Okay, thank you for that. So another question we have is, does the ATP NutriScan use spectroscopy for its soil testing? Or what examples are available for producers to use this technology for soil sampling?
I don't know about that ATP NutriScan you said. I'll have to look at if they could send me an email, I'll follow up on that and get back to them. I know it's not the greatest, most successful example, but I know that Farmer's Edge used to do what they called a virtual soil test. And that was the underpinnings of that was every sample that came in, they did near IR spectroscopy on, and then they were building models to try to estimate for fertility with the idea that you could do it much cheaper. I don't think that worked especially well at the time. But I think what we'll see are these sort of steps where you might not want to pay the money to test for soil carbon in your normal routine soil test, but because this works so well and it's so inexpensive that it'd be smart for labs to start to measure it as well. And I think that's where we'll start to see inroads much more before nutrient analysis.
Okay. So another question is, what do results from soil sampling using spectroscopy look like? These graphs look difficult to interpret.
At the end of the day, it's going to be a number. Even still, it's going to be what percent organic carbon is in that sample, what the total nitrogen is, it's really just the graphs and the figures are just kind of there to give you an idea about how close to that one-to-one line that the current state of the technique actually is.
Okay, thank you.
That's probably what's different about spectroscopists versus salespeople is when it works, once it works, it's a black box and it just gives you a number. And so my research program, my career is really about how much you can trust that number and how to make it better. And so a lot of times we spend time showing you the spectrum or showing you the scatter so that you can kind of evaluate whether it's going to work for you or not.
Okay, thank you for that. That is good information. So, do you predict in the future there might be training available for agronomists to better understand spectroscopy used in a field setting?
I do. I think that along with how to manage big data and how to handle GIS and GPS, I do think that it's going to be there, and I also think there's going to be commercial products that integrate a lot more with like the machines that we're pulling through the fields too.
Okay. Thank you, Derek. So another question we have is operationalizing large-scale soil spectra data involving development of soil spectral libraries that can be shared among users and projects might help in advancing this technique for agronomic recommendations. What is your view on developing such a project in Canada beyond soil carbon?
Yeah, I mean, I'm focused on soil carbon right now because it kind of works. But so what I think is that, well, I shouldn't. What I think is that we're probably going to see in the very short term, people turn to mid-infrared because there's peaks for phosphorus and sulfur that you can directly see, and the linkages between minerals and fertility are a little more clear in the mid-infrared. And you can buy auto samplers and you can run hundreds to thousands of samples in a 24-hour period. And there are a few labs across the country that are investing in that are academic labs as well as soil testing labs. And so I think over the short term that those databases and those data sets are going to be generated in Canada. And I think it's really important. I'm concerned a little bit about whether they're freely available for people to use and to mine and to interpret. I know that Microsoft and Google are investing a lot of money in agriculture and spectroscopy, and I don't think it's so that they can make all of that resources publicly available. So, my personal opinion is that we've collected data. It comes from the government of Saskatchewan. Some of the funding, it came from NSERC, most of the rest of it. And so it should be shared and it should be available for everybody to use.
Okay. Well, thank you very much for your questions, Derek. Very glad to have you here today.
Yeah, it was fun.
Thanks again. All right.